This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology. This is episode 429, recorded on February 16th, 2017. This episode of TWiV is brought to you by Blue Apron. Blue Apron is the #1 fresh ingredient and recipe delivery service in the country. See what's on the menu this week and get your first 3 meals free with free shipping by going to blueapron.com/TWiV. Hi, everybody. I'm Vincent Rackaniello, and you're listening to the podcast all about viruses. Today, I'm coming to you from Madison, Wisconsin. We're at the University of Wisconsin. I'm back. I've been here a couple of times. I was here last in November of 2013. That was TWiV 260. It was called Badgers Go Viral. And we'll be back again this summer, actually. We're here for the American Society for Virology meeting. We'll do another TWiV there. And in fact, the entire TWiV team will be with me. Dave Pommier, Kathy Spindler, Rich Condit, Alan Dove, and myself, and our special guest from the meeting. So you should check it out. Anyway, we have actually a few returning guests from the last episode we recorded here. On my left, Professor in Pathology and Laboratory Medicine, David O'Connor. Welcome back.
Great to be here.
And I have to tell people this. The last time you were on TWiV, you told me you discovered it because you needed something to put you to sleep at night.
That was true back then, and I have to say it's still on my phone today.
Does it still put you to sleep?
With some Ambien, I have to say I've listened and re-listened to the same episode several times on a long overnight flight to Brazil.
Yes.
And I hope that by the third or fourth time through, I've learned a little bit.
Whose voice in particular puts you to sleep?
Oh, well, I wouldn't want to say.
Okay. Well, thanks for coming back. We appreciate it. We also have another returned guest. He's an associate professor in the Department of Pathobiological Sciences, Tom Friedrich, all the way at the end.
Hi, Vincent.
Welcome back.
Thank you.
So here's the funny story about Tom. Has anyone seen that last episode? Halfway through, he left. So I turned to ask him a question, and I saw his mic sitting on the table, and he had left for a good reason. But I'll never forget it. He had to go pick up his child, right?
I was going to get fined if I stayed one more minute.
It's fine.
So.
It's totally fine. And I thought that was so funny. I think you're the only person ever to leave in the middle of TWiV.
Good to be famous for something.
That's quite an honor, I think. All right, we have 2 brand new guests on TWiV here today with us. To David's left, she is an MD, PhD, pediatric infectious disease fellow here at the University of Wisconsin Hospitals and Clinic. Emma Moore, welcome.
Thank you. Good to be here.
And Emma extended the invitation to come in the first place.
Yes.
Right, to do a seminar and then to do a TWiV.
That is correct. So he will be giving a seminar at noon tomorrow in one of our academic buildings on campus.
You should
which is
come,
wonderful.
because I'm going to talk a little bit about Zika.
I'm planning on it.
Okay. You might be interested, just a little bit. And finally, an OB, an obstetrician, right? Or
yes.
An obstetrician/gynecologist. Do they go together?
Well, it's a residency in obstetrics and gynecology, but I've also done a fellowship in maternal fetal medicine. So I'm a specialist.
You're a resident at the moment.
No, I'm a assistant professor.
Professor, but you did a residency in OBGYN.
Exactly.
Got it. And you are here at the University of Wisconsin Hospitals and Clinics.
Exactly.
Katie Anthony, welcome.
Yes, thank you so much for having me.
Yeah. And all these individuals are here for a special reason, right? And that's a bar of soap called ZEST, right? No?
Well, we need to have some acronym.
So I learned from Emma that there's something called ZEST here, Zika Experimental Science Team. And of course, as you would expect, you call it Team ZEST, which is redundant, right? You realize
Right.
we're going to point out all the issues.
Please.
Right.
Team ZEST, which we'll learn about. So you're all members of Team ZEST, right? And there are many others as well.
Many others.
And Z is the Zika part, right?
Yes.
Which we're going to hear about. So we're going to find out all about the genesis of it, what it does, some of your science, and that'll take up a nice episode. But first, 30 seconds, remind us of your background and training.
Sure. So I did my undergraduate at the University of Illinois, Urbana-Champaign. Then I came to Madison to do my PhD after not knowing really what I wanted to do with my life, except that I really thought infectious diseases were cool. And I joined David Watkins's laboratory, where I studied T-cell escape in HIV/SIV infections. And I've been here ever since. So I came to Madison in 1997 and stayed on as a postdoc, a scientist, an assistant professor, an associate professor, full professor, and then the problem, of course, is that the next stop is emeritus. And so you realize that there's really, you always have a step up that's a thing that's coming next until you no longer do. And then figuring that out was a bit of a life-altering thought, thinking, oh my God, there's nothing else here. And then, how about that, a fire alarm? See, there's always going to be something else here.
Is that a fire alarm?
Yeah, I think so.
It's period.
You can see we don't spend much time in the physics bowl.
Okay. I think it's funny that you say you thought infectious diseases were really cool, right? And I once told my class, I teach undergraduates at Columbia, I thought HIV was really cool because of its genesis from SIV, and all that. And a guy wrote, they have this website where they criticize the professors. You probably have one here too, right? And
I had a chili pepper on it once.
is that
Dave.
good?
I don't know. I mean,
You know what that means.
What does it mean? It's hard?
It means you're hot.
You're hot? Mm-hmm. Well, cool. Well, anyway, the guy wrote that I was morbid because I liked HIV. So I have to be careful what you say, right? Tom, tell us again in brief. The reason I say in brief is because if you go back to TWiV 260, we had you do the same thing. Tell us your background again.
Sure. Well, I'm originally from Milwaukee, and I came here in 1992 to Madison to be an undergrad student. I majored in molecular biology here. I actually worked for Ann Palmenberg as an undergrad.
Cool.
And then my trajectory is similar to Dave's. I trained for my PhD with David Watkins. I thought that HIV and SIV, like you, were very cool and wanted to understand their evolution, especially how they evade the immune defenses of each individual host, how they adapt to the host. And that really is kind of set the stage for the research I do today as an associate professor here at Madison.
All right. A lot of great virology here. In fact, Ann Palmenberg, of course, I did a TWiV with her a couple of years ago. It was called Right Form, Wrong Professor. Great story. If you don't know what I'm talking about, listen to it. The title was actually suggested by someone who was in the audience. He emailed it to me, and it's perfect. Because if you know how she got here, you probably know the story, right? It's a good story. All right, now we have two new guests we can hear about. Emma, tell us, where are you from originally? You from Illinois, Indiana, Iowa?
A little bit different. I was born in Australia, actually.
Australia?
Yes.
Wow, you have no accent at all.
I moved here when I was 10. So
It's
when
still
kids
a
make
long
fun,
time.
right? When kids make fun of you on the
Ah,
playground,
get
though,
rid of
for
it.
saying things differently.
Where in Australia? Big country.
I was born in Adelaide. And then I spent a lot of my childhood in Papua New Guinea, where my parents were missionaries.
Wow.
So after that, I moved to Wisconsin at 10.
Famous country for kuru.
Yes.
You know the story, right?
Yes.
Carlton Gaidusak and all that.
We were not in that part of the
No, I
country.
understand. But that's the only thing I know about Papua New Guinea.
Yeah.
But it's a great story.
Yeah.
You lived there for how many years?
8 years.
18?
8 years, sorry.
No,
I moved-
but you're 18 years old by the time you leave, right? Because 10
No.
years in Australia.
So I was 10 years when I moved to the U.S. I was
Okay.
2 when I moved to New Guinea.
Okay, and where did you move to in the U.S.?
Wisconsin.
Okay, and you went to high school here?
Yep, high school here. I went to undergrad at Gustavus Adolphus in Minnesota.
Okay.
And then I went to the combined MD PhD program at the University of Iowa, their medical scientist training program. And then from there, I went to do a pediatrics residency program at Emory in Atlanta. And then after that 3-year residency, I came here for fellowship. And so I've been here for a year and a half.
So you're a pediatric infectious disease physician, correct?
Correct.
All right, so where did that interest come from? Were you, as a young kid, were you interested in science or being a doctor or both?
I think I have always been interested in infections and diseases related to infections. It may stem from my upbringing in foreign countries. And I guess from the beginning, I didn't know if I wanted to do infections from the clinical or the research route, and I couldn't figure that out or tease that apart, so I decided to do both.
Okay.
And then I knew I wanted to do infections as a specialty. I just didn't know if I wanted to do adult or pediatrics. So I went the pediatric route.
So for our listeners, what does it mean you're an infectious diseases fellow?
Right.
Are you a doctor, a scientist, or both, or what is it?
So I'm a doctor. So after people graduate medical school, they go to residency to learn their medical craft. So I am a full-fledged pediatrician doing subspecialty training in pediatric infectious diseases. So this fellowship program is 3 years long, and after that I will be a pediatric infectious disease physician. Right now I see kids with complicated infections in the hospital and in clinic as well.
Are you going to stay here, you think, after you're done with your fellowship?
I would love to.
And you'd be a staff physician, for example, that would be the idea. Would you like to also do research?
Yes. I hope the majority of my time will be doing research.
Okay. Now, as a for your PhD part of your training, what did you work on?
I worked with Jack Stapleton. We were studying GBVC and HIV co-infection.
This was in Iowa.
At Iowa.
Iowa, right. I know the name very well, yeah. And here in the lab, do you get a chance to do some pipetting? Do they let you?
Yes, they do. I don't do as much as other people because I have, random duties away from the lab, but I do help process many of our Zika samples. I shouldn't say many. Other people do a lot more than I do. But when I am there and on some weekends, I can help process our Zika samples in the lab.
So here in Madison, Wisconsin, what is the most common childhood infection that you see?
Let's see. There are a few bread and butter ones that we see a lot. So the most common one we see in kids in the hospital would be bone infections, so osteomyelitis or septic arthritis, a joint infection. It's probably our most common one.
Is that typical for most of the country, or is it specific to here? Is it a rural type of thing, or?
I would say that's pretty common across the country.
Interesting.
So, there are lots of infections that regular pediatricians who don't have specialty training can handle on their own. But when it gets to this with, IV antibiotics for perhaps a couple weeks, we help manage the patients after they get home from the hospital. So we really see those patients.
So you see children who come in with serious problems that primary care physicians can't handle, essentially, right? They don't. Right. I don't know what this is, or I need help.
Yeah.
Got it. So common colds, they stay in the in the primary care, right?
I didn't want to see those every day.
Well, especially because there's not much you can do, right?
Right.
Okay, except as Anne would say, have chicken soup, warm chicken soup.
True.
So Katie, where are you from?
I'm originally from Wisconsin. I'm from sort of a town between here and Milwaukee. It's called Heartland. And that's where I grew up and went to high school and everything. I went to college at WashU in St. Louis. I did my medical school in upstate New York at University of Rochester. And then I did my residency and fellowship at Baylor College of Medicine in Houston, Texas. So
great places.
It is.
So that's a doctor I would pick based on. I just had to pick a new primary care physician. I'm looking through the lists, right?
Oh, yes.
And I see a guy, he went to UNC, University of North Carolina. He went to SUNY Buffalo for medical school. Can't remember the residency, what it was. So I told him, I picked you because of your pedigree. It's important, right? It's important to do. So you're an OBGYN at the moment, right? And you also specialize in maternal fetal care. Is that correct? Got it written down.
Maternal fetal medicine.
Medicine. So what do you, as an OBGYN, what do you do?
So after completing a residency in obstetrics and gynecology, you get boarded to provide care in both obstetrics and gynecology. And so obstetrics is specifically care for women who are pregnant, whereas gynecology is sort of more care surrounding, the uterus and the pelvic organs, hysterectomies, abnormal uterine bleeding, that sort of thing.
Okay.
By pursuing a fellowship in maternal fetal medicine, now I pretty exclusively see women who are pregnant, and specifically women who are having high-risk pregnancies. And so while I am qualified to do, provide care for, general gynecologic care, now I exclusively see women who have high-risk pregnancies.
So you spend most of your time looking at patients, right? In contrast to Emma, who spends a little time doing experiments, right?
Right. I spend about 70% of my time providing clinical care, and the rest is for research. So it's a smaller percentage.
So I think as we explore ZEST, we'll find out how you brought this team together and your contribution to it. So when did ZEST begin, and whose idea was it? Was it your idea?
Well, I think last year you talked with Esper Callis.
I did.
He's one of my close colleagues from the University of Sao Paulo.
Right.
So we've had a long and productive collaborative relationship with scientists in Brazil. And in November of 2015, they told us about babies being born with small heads in the northeast of the country. And right around the same time, Jorge Osorio, who's a professor here on campus in Tom's department, had been studying Zika infections in Colombia. And so I was telling Tom about my trip to Brazil, and Jorge was telling Tom about his observations in Colombia. And I think Tom said, well, we really should probably all be talking about this together. And that was really the genesis of what became ZEST, because we quickly realized that there was complementary expertise. I knew nothing about mosquitoes. No one in my lab knew anything about mosquitoes. But we had some specific expertise, especially in the ability to study experimentally infected non-human primates that Jorge's lab didn't really have. And so we got together with Jorge's lab, and especially Dr. Matt Aliota, who's here today, who's a scientist in Jorge's lab who has a ton of experience studying arboviruses. And we began to put together a plan to try to develop an animal model for Zika virus. And this was back in November, December 2015. For the next month or two, we were aggressively trying to figure out how we would do this. We already had a bit of an advantage because in a completely independent line of research, we had been studying viruses that we had found in wild African primates. I think we talked about this the last time you were here. And that required a very extensive risk assessment with biosafety and with the primate center, because we were taking plasma from baboons that was collected 35 years ago, and we were injecting it into macaque monkeys in the lab. And we had made sure that that plasma didn't have any viruses in it other than the virus, but we could never be sure of what was going to be happening. There was an element of uncertainty in those experiments. But having overcome that with the viruses, we were in a good position to propose similar experiments with Zika virus, not knowing exactly what would happen to the monkeys that were infected with Zika virus, and more importantly, trying to make sure that caretakers and veterinarians and people who would be working with the animals on a day-to-day basis would be informed about the potential risks that they would encounter, and taking all the precautions necessary to make sure that those risks were minimized. So having done that, we brought in the Primate Center and their expertise in animal care and veterinary care, and then we quickly realized that if the issue here was going to be Zika virus during pregnancy, neither Jorge's lab nor my lab or Tom's lab had any expertise at all in congenital infections. So Sally Permar from Duke University, who's an HIV and cytomegalovirus researcher, contacted me independently in mid-December and said, hey, have you heard about this Zika virus? Maybe we should do something with that. So we then started to put together a team where either people would reach out to us or we would reach out to them. And we really tried to capture expertise in all of the disciplines that would be necessary to study a new congenital infection that we knew very little about. And one of the great things about the University of Wisconsin Madison is how many different types of specialists we have in our vet school, our med school, our grad school, our nursing school. And we would contact people and say, hey, have you heard about Zika? I'm in the pathology department. Can you help us? And to a person, people said, yes, we'd be happy to. And so we brought together an eclectic, large team of investigators from around campus, and if someone on campus didn't have the right expertise, I remember in particular we talked to one neuropathologist who didn't really think he had the right sort of neuropathology expertise. He would refer us to one of his colleagues somewhere else who he thought did have the right expertise. And so by a year ago, Andrea Whaler, who's a member of our team, reminded me just before I came over here that we infected our first animals a year ago yesterday. Right, it doesn't seem like a year to those who were here.
The day after Valentine's Day.
But it was by that time we already had about 30 people or 40 people who were involved in the project directly. So it became a big team, and we realized that a big team needed a clever acronym. So we came up with ZEST, and that was how we got here.
So how many people are in the team now, total?
God, I don't even know. I think our Slack channel has about 40 members.
Okay.
Or something like that. I think it's also important to mention the contributions of Ted Golos, who's a maternal fetal medicine expert, and had expertise, was affiliated with the Primate Center and had expertise in congenital infections in the setting of listeria, not a virus, but nonetheless, it was this sort of core of expertise made it possible for us to get Team ZEST off the ground really quickly here at UW.
So Emma, you were already here, right?
Yes.
And what did Dave say? You want to be part of ZEST?
Well, I was sort of just joining his lab at that point, and he said, "Hey, do you want to write an animal protocol?" And I said, "What's that?" So
That should be it for a while.
Yeah. So that was my introduction.
Okay.
Figuring out how to do that.
So as a pediatric infectious disease expert, that was your contribution. That is your contribution to the project, infections of young mammals.
Yeah. So I mean, as a pediatrician, I really like to think about, how is the fetus developing? What sort of problems is the fetus getting from this viral infection? And then are there ways that we can mitigate this and interrupt this problem?
And Katie, you were brought in as the expert in pregnancy, except it's not human subjects, it's non-human primates.
Right. And so obviously there's not necessarily an exact translation, but I've been able to assist with interpreting the ultrasounds, performing I bring over one of our sonographers, who assists with the ultrasounds, and I also perform the amniocenteses. So
you had to learn how different it was and how to interpret things. Did you have a veterinarian to help you, or was that not available?
I mostly actually did lit searches, and we've also been contacting an expert in Oregon, Alice Trontel, who, sort of did a lot of the nomograms and was one of the early people who got on board on performing ultrasounds in rhesus macaques.
Okay. So
fetal rhesus macaques.
Right. Let's talk a little bit about the experiments. You've published a couple of papers so far. You've also put all the data online. Let's talk first about that. You had this model where whatever you did at a daily basis went to a website. What was the thinking behind that?
Well, again, it stemmed from this sprawling team that we had about a year ago when we started these studies. So even at that point, we realized that there was going to be a large number of people who were going to be interested in our results. And so I remember Tom and I were talking in his office about how we could keep everyone who we had contacted, who had provided guidance on how we would design the experiments and what we should be looking for, how we would keep everyone in the loop. And we decided that we would put our results on a web portal that was powered by the open source LabKey server platform. We'd been running a LabKey server in our lab to manage purchases and experiments and freezer inventories for a number of years. And so we already had experience with the platform. And we said, well, we can just put the data there and give access to everyone on our team. And then at some point, the conversation morphed into, well, if we're going to be among the first to have non-human primate data, and we know that other people are also going to be developing animal models for Zika virus, because by this point, you already had infections in the US and you had the beginning of this tidal wave of people doing research on it. If we're going to be among the first, whatever we learn is going to be useful to other people too. What happens if instead of restricting the access to people in our team, we just made it open to everyone? And I honestly don't think we thought about it much more than that. We realized that there would potentially be some negative implications from doing it, but we, didn't think about it too hard.
What did you think of the negatives?
Well, I think at the time there was a concern that animal rights activists might not look kindly on having data made available. There's, we're subject to FOIA requests fairly regularly. There's the concern that we were going to put up information that could be wrong and misleading. That's still a concern. there was a concern that we were going to compromise our ability to eventually publish the work. I don't know, Tom, are there others that I'm forgetting?
No, I think that's it, and I think especially we had that latter concern for the more junior members of the team who were going to be using this to kind of establish themselves, and there's a real concern in when we think about making data publicly available pre-publication. Does that open us up to the potential for being scooped? Does that reduce the potential impact of whatever publication might come out of it? But in the end, I think we felt that we briefly considered some kind of model where people might register and have free access, and we just sort of have some sense of who they were. But we decided in the end that was too much trouble, and we might as well just make the data completely available to the public. And there have really been very few downsides, I have to say. And we thought an upside that we haven't discussed so far was that, as Dave said, not only were we going to be among the first to infect monkeys with Zika, and a lot of other researchers would be interested, but we felt if we do something that's wrong, we can make a note of that in the record, and that's valuable information. So, in a situation where the epidemic is evolving rapidly and research is evolving rapidly, there's no good avenue for communicating negative results. Like, let's say this viral strain doesn't work, or it has, there's something wrong with it. Rather than allow other people at other institutions to find that out for themselves and reinvent the wheel, we felt they should learn from whatever mistakes we will inevitably make when we start doing this in a new area of investigation. And that was, I think that kind of tipped the balance to just make everything available.
Has anyone else doing any kind of Zika done similar thing?
I think there's been people who are interested in it. Certainly a lot of people are following our model closely. I think a few people took very tentative steps towards it, but I don't know how many people are keeping up with it currently.
There's not as much from the other primate centers as we've been able to place, and I think partially we have an advantage because, as Dave said, we have experience with this platform, and it's relatively straightforward for us to do.
I think it's a great idea to have open data, open science, right? But as you know, it's you're kind of an advantage. There are not many people infecting primates with Zika virus in the world. So I've seen a couple of other groups publish papers on it. So you're not going to get scooped. But if you move down a little bit to molecular biology experiments, there it's harder. You publish your thing and someone can say, oh, look at this approach. We have to do this. And it's unfortunate that it has to be that way, but it is.
Well, it sounds a little preachy, but it's also true that what we would call getting scooped is also called advancing knowledge more quickly. If someone else gets there faster, then the knowledge gets out there faster, and in the course of a rapidly evolving epidemic, that's still a net good. It might not be good for the person who was scooped, but it's good for moving the knowledge forward. And there's also the element of who we do the work for and at the behest of. So if we're going to NIH or Gates Foundation or whoever it is saying, we need this money urgently because it's an emergency, the money is given to advance the knowledge as quickly as possible, not to advance the scientists who receive the money to do the experiments and then publish them 6 months later in a prestige journal of their choice. So, we often say that we're stewards of taxpayer money. And so getting information out there as quickly as possible is responsible stewardship.
I totally agree with you, but you have a tenured position. And there are a lot of people out there who want to have tenured positions, and they don't want to be scooped. So this is a problem. For high priority things where we have an outbreak and we need to make a vaccine or an antiviral, I totally understand the sharing. But at lower level investigations where maybe the results are not going to provide a cure or a therapeutic, people have to have careers, and we have to balance that. Basically, it's not so easy, right?
Well, that's a question of changing the system, though. I think that
we're not going to do that today.
Well, I don't know how much time you have.
Is that on the agenda?
No, I agree. It's a big issue. Publishing and all of this is a big issue, but I think you have a really it's great that you did this, but you have a special situation where you have a system that few people have. It's an emerging situation where you got money specifically to do this, and I totally agree that the results need to be shared. But on many other levels, at the moment, our system just doesn't support that, which I lament totally.
I remember your discussions about preprints in mid last year.
I mean, I'm in the middle of this myself. I totally support preprint servers, open access. I think the journals are screwing us all over, the high-profile journals, but we use them because we need to get jobs, we need to get grants. And my lab is in the same situation. We're about to submit a Zika paper, and my person, she wants a high profile so she can get a job. And I've been talking about this and writing, and people are saying, how could you do that? You're an open access preprint person. I said, I know, but she needs a job and I don't know what to do. I can't screw her career over because of my ideals. So it's very difficult, as you know. But that's not what we're here to talk about today. We're not here for me to angst over this. Let's talk about your experiments. So the initial choice of the animal, you had been working with non-human primates here, so that was what guided it, I presume?
Yeah, I'll let these guys talk about that.
We can talk from a pregnancy perspective in a second, and so I'll just say a couple things about it. The main issue for us was that this was apparently a congenital infection that was causing birth defects, and so if we were going to have a model system that would help us understand how a viral infection would cross the placenta and do something to the fetus to cause birth defects, and that was the main pathogenic outcome, then we needed a model that would sort of faithfully recapitulate human pregnancy. And so that really kind of took rodent models and small animal models off the table, because the physiology of pregnancy is just fundamentally different in those models, and that is the extent of my knowledge about pregnancy. So I will turn it over to the actual experts.
So what is the actual animal that you use in your experiments?
So we use rhesus macaques.
Rhesus macaques.
Yes.
These are old world monkeys or new world monkeys?
Indian origin rhesus macaques.
So they're new world.
Old world.
They're old world. Indian and African are old world.
Yeah.
Yes.
South American or New World.
Got it. So give me an example. Are there any macaques that are new world monkeys or not?
Not macaques, but marmosets, tamarins.
Okay. So you picked an old world species because you have them here, but it's good because this is an old world virus originally, presumably, right?
Well, the export of Indian rhesus macaques from India was banned back in 1978.
Right.
So there are 7 national primate research centers who have a mandate to maintain breeding colonies of these. So the breeding colony system means that we have a lot of expertise with pregnant Indian rhesus macaques, which is why we chose that.
And this center is here in Madison, this primate center?
One of them is, the Wisconsin National Primate Research Center.
Where they're bred and use them for your experiments as well. So tell me, what's the difference? The pigtail macaque is a different species?
Yes.
Also an old world?
Yes.
Also an old world,
But I've seen some papers with those, not from you, from other groups, correct? Right. All right, and cynomolgus macaques are another species?
Yes.
Right, those used to be used for polio. Albert Sabin used many cynomolgus macaques.
That's right, and they're still used. We use cynomolgus macaques for a lot of our SIV research, especially those from the island of Mauritius, because they have greatly reduced genetic diversity compared to the rhesus macaques.
Okay.
Their founder population.
But rhesus were here, and as Katie mentioned before, there's a pretty well-established kind of growth trajectory for the development of the rhesus fetus, and that's kind of what we needed to assess pathogenesis in this model.
What's the gestation period for a rhesus?
165 days.
How many months is that? 4 months? 4 times 3 is 120. 4 times 5 times
about
5.5
something.
months.
So shorter than humans, obviously, right? Okay, but still.
We can do math.
I've always looked at it at days, but
Yeah. So, but it's still long enough to get trimesters, whereas in
Exactly.
a mouse, a trimester is over before you know
Yeah.
it. That's the problem, right?
yeah, there's predetermined, trimester cut points, and
Okay.
so.
All right. So, what was the first experiment you did? Does anyone remember?
ZICV-01.
What did you do?
So we infected 3 rhesus macaques, non-pregnant. I think it was with the African lineage Zika virus.
No.
That was Asian lineage, so
Yes.
French Polynesia. Okay. So we infected 3 rhesus macaques with French Polynesia Zika virus.
Why did you pick that virus? That isolate?
That was this very similar in sequence to the strain that was going around South America.
Okay.
and we did not have access to all the strains that are available now.
Okay. So what route did you inoculate the animals?
Subcutaneously.
Okay. Where? Arm, foot, leg?
Cranial dorsum, if I recall.
Yeah.
This-
And so subcutaneously mimicking a mosquito bite in some
That's
way.
what we were trying to do.
Yeah.
Yeah.
But you say trying, but you don't think that accomplished it?
No, as good of a model as it
Got
can.
it.
We
Okay.
have done more experiments now to actually see if that's the case.
So how much virus did you put in? How did you know how much virus to put in?
Well, we had to speak with our arbovirologist like Matt Aliotta over there and say, hey, what does a mosquito inject into the skin?
Yeah.
And so, they went back to the literature looking at West Nile and dengue and said, usually we think it's, 10 to the 4th, maybe 10 to the 5th or 10 to the 6th. That'd be really high. So we decided to try 3 different doses.
It was actually Kristen Bernard, another virologist here at UW-Madison, who had established that sort of dose range that's delivered by a mosquito in the setting of West Nile. And so it was unknown for Zika, but we just used that as a model.
Okay. So you took 3 animals, male, females, what, mixed?
I think they were mixed. Males. I'm getting males
Okay.
from the audience.
Thanks, audience.
All boys.
You infected
Thanks.
them, and then what did you do? How did you monitor them?
So we did the same sort of arrangement we do nowadays. So we look for daily 10 days, doing daily blood draws, getting saliva, getting urine, looking for virus in all those samples. And then twice a week or weekly after that for 28 days.
Did you also look for fever, elevation of temperature?
Yes, our veterinarians looked for that. I don't think we ever saw fever. Is that right, audience? Okay.
All right, so what did you find?
So we found virus in the blood and the urine.
How soon after inoculation?
Usually by day 2, we saw pretty high levels in the blood. So it peaked between days, I think day 2 and day 3, and then slowly came down to not detectable in these non-pregnant animals by 10 days or so.
Okay, so that was the first experiment. It's an acute infection, peak, and all this is measured by PCR, right?
Right.
So why didn't you do plaque assays originally, and don't seem to do very many still, because I can read from your papers.
So we do plaque assays. They are a lot more time consuming, and with the number of samples we get, we choose to do them on ones that we're really interested in. And that Matt Aliotta does that for our lab as well. So he does the plaque assays. So we do have plaque assays from our first paper showing that we have, live virus in the, I can't remember, was it serum or plasma that you looked at? Serum.
There's a figure there at the bottom, yeah. It's very low titer. It's like 10 to the 2 PFU peak per mil of serum, right? Why do you think it's so low? It seems to me that would be hard for a mosquito, if this is a monkey in the forest and mosquitoes are transmitting among monkeys, that might not be high enough to sustain transmission, right?
I don't know the reason for that exactly.
I'm sure you've thought about it though, right?
I know that. So, I mean, the live virus only detects live virus. PCR can detect just fragments in the blood if there is any. So that's probably why that's higher. I don't know. So I know plaque assays, many groups around the country have found that we do get very low titers. We don't get very high titers when we measure it. So maybe it is that low, or maybe there is, a different assay that we should be looking at for live virus.
Well, there's nothing better than a plaque assay. It's the best assay ever devised to humanity. And I will argue with anyone forever. There's nothing better because it detects infectious virus. Now, it does have a limit, of course. If you're putting 0.1 ml onto a monolayer, you have to have 10 PFU per mil. Otherwise, you're not gonna see it. But there is a paper from, I think it's Michael Gale is on the paper from the Seattle primate group, where they say in none of their experiments could they detect infectious virus. They only found signals by PCR in their non-human primate experiments. So there's something going on which certainly merits investigation, right?
Well, some of it also might be the samples that we use for the plaque assays. If I remember correctly, we sent you some samples from our first infected animals because you wanted to do plaque assays with them.
That's right.
And you wrote me back a week or so later and said, what did you send me? This garbage has no virus in it. And it turned out that was because we sent you plasma, I think, and we discovered that you really can only plaque out of serum, not out of plasma. So whatever we're measuring, we have to be open to the possibility that we're underestimating the actual amount of replication competent virus because of the way that we're giving you samples.
It's also probably true that I mean, as Emma already suggested, that PCR does detect non-infectious particles, and there's probably a vast excess of non-infectious particles over infectious particles. And so, we've found a pretty consistent relationship between the amount of viral RNA genome copies that we detect and the number of infectious units that we detect, where it's about 1,000 to 1 ratio.
There's a paper just come out this week from CDC where they're looking at a sexual transmission model in mice, and there's a lovely graph where they do PCR and plaque assay. They infect males and then they look at virus in semen, and you can see RNA for 5 weeks longer than infectious virus from semen alone. So something, and they don't know, they speculate that it might be neutralized virus or something, but clearly when you're first establishing a model, you need to do both assays to know if this signal means virus or just a fragment. So now we know in mice, past 21 days, there's no infectious virus. There's just, and that's important because, in the human studies, people say, this person excretes virus in semen for 6 months, but it's not virus, it's PCR products. I think you have to be really careful about that. Anyway, these initial animals that you infected, acute infection, then the virus was cleared, apparently. So what was next? Were you excited about that? Did you think, here we have a model now?
No, I mean, we needed to look at other aspects too. So were these monkeys protected from re-challenge with the same strain or with a different strain? So that was another paper that we did. So we infect 2 groups, one with Asian and one with African lineage Zika virus, and then challenge them with the homologous or heterologous virus. So re-challenged them both with Asian lineage Zika virus and found that they were protected from reinfection. And that was Matt Aliota's paper.
So when you initially infect these animals, they have an acute phase, and then you've measured antibodies in them, I think, right? Neutralizing antibodies, they have also a cellular response. And then you challenge them, and there's no viremia. Is that's the assay? Because they don't show any disease, so you can't use that as a...
And critically, no anamnestic antibody response.
Meaning?
We didn't really see a boost in the antibodies after the re-challenge, suggesting that it was really a sterilizing immunity, not a localized reinfection that just didn't make it out into the periphery.
Right, so every animal you challenge is protected from these experiments. Right?
Small n, but yeah.
So has anyone ever criticized that? You can't use a lot of animals, for obvious reasons. They're expensive and so forth. Is that a problem at any level?
Yes. It's a consistent challenge because the sample sizes in non-human primates are going to be small, which makes powering studies appropriately very difficult. So we have to be very, very careful against over-interpreting our results. And when you get into smaller slices of non-human primates that are available, so for example, instead of being able to use Indian rhesus macaques, you have to use pregnant Indian rhesus macaques. The more narrow your requirements are for the animals that are available, the fewer animals you're going to be able to use. And so for the pregnancy studies, we've been really limited in terms of how many animals we can use. But what we try to do is get as much information as possible in as many dimensions as possible from every animal that we study, in hopes that even if we can't get a sufficiently powered analysis by using lots of animals, we can get as much information as possible from each animal that we can study.
So in one paper that you published, you had infected some pregnant animals, and you said, we're following these up, and that was a long time ago, so I presume that's done. Can you tell us what happened to those animals? So these are infected at what, first trimester, correct?
They were infected at different points. 2 of them were in the first trimester, 1 was in the second trimester, and 1 was in the third. And Emma can probably speak better to this, just sort of looking at the duration of viremia and...
Yeah, so it was interesting. In the pregnancies, we found a lot different natural history of the virus. So we found prolonged viremia in 3 of the 4 animals. So one animal that was infected in the first trimester had viremia detected by PCR in the plasma for 70 days after infection, which is, a lot different from our non-pregnant animals.
And that's similar to what we see in humans too, actually, just to correlate.
So that, lets us model that aspect of.
Exactly.
And what do you think is the reason for the persistence in a pregnant animal?
we've been talking a lot about that. That's a really great question. So it could be a couple different things. So I think our first thought was that, pregnant animals, even pregnant humans, we in the medical field always think of them as slightly immunocompromised because the fetus growing in you has different DNA, so you don't want to reject that fetus. So there's some sort of difference that happens in your immune response to that. So could that be because they aren't clearing the virus? That was our initial thought. I think now sort of what we think or suspect is that maybe there is a reservoir in the fetus that is actively allowing viral replication. And so that viral replication is leaving the fetus, going through the placenta and back into the mom. And that's because we see neutralizing antibody in the mother. So it looks like she's making a good enough antibody response, and she's the same sort of T cell activation profile that the non-pregnant animals do. So we don't know this. This is what some of our studies are examining, but that's one of our thoughts.
It could be in the placenta as well, right?
It could be, yeah.
Or of course, hard to separate the two because they go hand in hand, right?
Yeah.
What about any effect on the fetus? Have you seen anything?
So I think this is where Katie can really come in here.
Yes, and so what we did see in the fetus is we saw that sort of the growth trajectory of the head. It wasn't, whereas early on in pregnancy when we were looking, we were seeing that it was kind of tracking right along the mean. We saw it sort of fall off. It didn't meet the clinical definition of microcephaly that we use for humans, which is a head circumference measuring 3 standard deviations below the mean. But in 3 of the 4 animals, it was measuring between 2 and 3 standard deviations below the mean, and the other one was also below average. And so, so it wasn't technically meeting criteria for microcephaly, but it was looking suggestive. And then also, we don't know if we followed these, followed postnatally, whether microcephaly might fully develop later, which is something we've seen in humans.
Do we know that all 3 fetuses were infected?
Yes, we know that actually the vertical transmission rate to the pregnancies was 100% in all of the fetuses.
And I think you also said the fetuses became seropositive as well. Is that correct? Or am I thinking of another paper?
So when we looked at the fetal tissues, we saw infection in the placentas of all 4, and in 3 of the 4 fetuses we found virus by PCR in some of the tissues, and also we saw evidence of inflammation and infection on histopathology.
Did you let these go to term, or did you abort them beforehand?
A C-section was performed somewhere around 10-ish days, sort of on average before full term.
And then
which is similar to in humans,
And then you analyzed the fetuses for virus, and you did some histology, I presume, on different tissues, right? And so, where's the virus replicating in the fetus?
If I remember correctly, I think it was brain in some of the animals, some of the eye tissues specifically.
Optic nerve and whatnot.
Yep.
And that's where we're seeing a lot of
nodes.
inflammation as well, is in the eye.
But there's a question about this because we allowed the fetuses to come close to term, and there was this prolonged viremia that Emma talked about in 3 of the 4 pregnant dams. But even though that viremia was sustained, there was a long period of time between the clearance of viremia, even in the mother who had shed virus from blood the longest, and the delivery of the baby. So we don't know the extent to which infection might have been cleared in each of the fetuses before we were able to analyze them. So we know that there was viral RNA present in a collection of tissues in the fetus and then at the maternal-fetal interface in each of these pregnancies, but we don't know what we might have missed by collecting the fetus at term rather than right around the time that viremia was resolved in the mom, let's say.
Wouldn't it be nice to have a non-invasive way to monitor viremia in both the mother and the fetus in every tissue, an imaging-based method to look at virus?
Yes, absolutely.
Someday, a tricorder type thing. Yes. Someday it will be.
Someday.
But, so do you have plans to do these experiments with different isolates of Zika, for example, to see if there's any difference in their pathogenic potential?
So we're actually currently in the process of switching over from the French Polynesian isolate that we talked about earlier, and that has been kind of the subject of the papers we've published so far. that one, if you look at the phylogeny of Zika viruses, is ancestral to all the viruses that circulate in the Americas now. But there are some changes relative to that virus in viruses that are circulating now, and there's been some speculation that maybe we see microcephaly and other problems because there's been some specific mutation in the virus, and that allows it to invade the placenta or cause birth defects in some way. So we're now working with an isolate from Puerto Rico. And we've made a large stock of this virus, and we're hoping that this can serve as a resource not only to experiments that are being done here at Wisconsin, but to other investigators who want to use the non-human primate model. So maybe we can kind of standardize around, a recent isolate that has been pretty well characterized. So we don't know yet whether this virus will behave any differently in the setting of pregnancy or non-pregnancy. We've infected some animals with it here at the Primate Center so far, and the viral replication kinetics are very similar to what we've seen in our previous studies with other Zika viruses.
So that's a good point I wanted to bring up. There's the idea that either the virus sustained some mutations that allowed it to become more pathogenic, or it just happened that it was introduced into a non-immune population and took off, which we know happens with other viruses. And there's really no way to distinguish those two at the moment. Yet, if you read certain review articles, people are drawing to conclusions. Even the press will draw to conclusions. Do you have any? I mean, my feeling is that it's always had the capability to do what it's doing today. I don't see why, and I would love your thoughts on this. I don't know why a virus would evolve to infect a fetus. I don't think that helps it transmit to other hosts, right? First of all, not everyone is pregnant, and unless the virus can effectively get out of the fetus, maybe that happens. I don't see why that would be a selection for evolution. I think the virus has always been able to do it, and we just never saw it in the healthcare poor areas where the virus was circulating. Any thoughts?
Well, I think within the fetus, within one infected host, the virus may enjoy a selective advantage if it can replicate in some compartment that's available to it. So, within the host, there may be a selective advantage to, replicating someplace where you couldn't before. But I agree, in sort of the long term, there's no particular selective advantage to infecting the fetus.
Well, if there's a prolonged viremia, perhaps then there's a prolonged period of time that the host that it's replicating in, could serve as a reservoir, so to speak.
Yes, and so we've speculated that in our ZEST meetings, right? That maybe if there is this prolonged viremia, then maybe infected pregnant women could serve as spreaders of the virus in the community. I do think it's also possible that we and others have seen that immunity that's conferred by one infection or by vaccine constructs seems to be very potent again, and protects against re-challenge. So we saw the sterilizing immunity that Dave talked about. And if you're living in an endemic area, then it's and the attack rate is very high, then it's quite possible that most people are infected before they reach reproductive age, and then it's off the table to infect the fetus.
I think that one of the biggest knowledge gaps in Zika virus right now is that we have no real information about what the epidemic is looking like in Africa. There's some very limited data from 30 or 40 years ago, and as an HIV person, I found it remarkable that it was 3 years after the description of immunocompromised people in the US before there was a recognition in print that there was HIV in Africa. And I think here we know that Zika is in Africa because that's where it was discovered. But we don't have any data, to my knowledge, suggesting whether 100% or nearly 100% of people in affected communities have had Zika, whether it's a childhood infection, whether it's not, whether the same thing has been happening for decades in Africa, but in the backdrop of high infant mortality and other issues associated with childbirth, it simply hasn't been recognized.
Well, of course, the serological tests for Zika aren't great anyway, so it may not. You may not be able to distinguish between dengue and chick and Zika, and so we're not. And I'm sure we'll get to that point, and then we can do those studies. That would be brilliant.
Yeah, they're
Look
getting
at it.
better. I mean, we'll hopefully have an answer on this soon enough. But I think that we need to make sure that people are looking and are funded to look.
So I was going to ask you if you have projects that involve going to Africa and sampling primates, right? That's what we talked about last time.
Right.
Are you doing this for Zika, or not yet, or you would like to?
So we actually have a paper that's on bioRxiv and was just accepted by MSphere yesterday with Connor Buechler as the lead author, where we found that we went back to some of our old samples and we looked. We used our experimentally infected macaques to calibrate a serologic assay to overcome this issue of what a past exposure would look like. We had macaques with various types of Zika and dengue exposures. We then used that assay on old samples, and we found that there were Zika-specific antibodies in baboons and African green monkeys in our old sample collections. And we're now working with Scott Hensley and his team, who has access to samples from other populations in Africa to look at complex exposure histories to flaviviruses in some of their cohorts.
Right.
We're just starting those studies.
So Katie, as an expert, does it make sense to you that there could have been Zika-induced microcephaly at, say, low levels throughout Africa, even parts of Asia, and we just didn't see it?
Absolutely. And I do some global health work in Africa. And I mean, I don't know what it was like, back when this infectious disease was potentially emerging in the 60s and 70s, but the record keeping still isn't ideal.
Yeah.
A lot of people still deliver in huts in their villages. There's no records. And so I think it's entirely possible that, this has been causing congenital problems for half a century or even longer. And we just would have never known about it because either maybe there wasn't a big spike, or if there was a big spike in the cases of microcephaly, it wasn't recognized, I think.
Right. So going forward, what are the plans? Do you plan, for example, to look at sexual transmission in macaques, or is that too difficult to do? Who's going to answer that one?
Well, the actual model of sexual transmission in macaques would be very difficult. In fact, investigators here at Wisconsin have been looking to establish a sort of model of SIV sexual transmission, and that proved to be very difficult indeed.
So they don't mate? You have to artificially inseminate?
To control the inoculum and to understand what the dose is that you're applying, and especially for SIV, we know, and HIV, we know that the chance of successful transmission from the perspective of the virus in any particular instance, the efficiency of transmission is very low. So we don't have immediate plans to directly model sexual transmission in macaques, but we actually have been interested in understanding the potential for non-vector mediated transmission. So we have a study that we did over the summer and fall that we're just getting ready to submit. Actually, is just been submitted. That was led by Christina Newman, a scientist in Dave's lab, looking at sort of mucosal transmission. So you may be aware that there was one example of non-vector, non-sexual transmission that was reported out of Utah, and there are anecdotal cases in addition to that. So we tried to assess, what would be the potential for contact transmission. So we applied concentrated viral stock directly to the tonsils of 3 macaques, and they were all productively infected at a dose of 10 to the 6th PFU. And then we infected a series of donor animals subcutaneously with 10 to the 4th PFU, as we've done for all these other studies, and then tried to collect their saliva. So we had vet staff carefully pipetting saliva out of the mouth of an anesthetized monkey, and then applied that to the tonsils or nasal mucosae or the conjunctiva of naive recipient animals, and in no cases did we detect transmission where we were evaluating the actual saliva from an infected animal. And we found that the amount of infectious virus in that saliva was extremely low. So our conclusions from all of that are that mucosal transmission, or sort of more casual contact transmission, is a theoretical possibility, but it would probably require that the source host have a much higher level of virus shedding than is typical.
But it's been found in human saliva, though, right?
Yes.
The virus.
Yes.
For sure. So I've always wondered, how can you rule that out in these supposed cases of sexual transmission? You don't know, right? It could even be, skin lesions that have blood, virus-containing blood. But it's interesting. So if you infect a macaque subcutaneously, do you find virus in saliva?
Yes, we do. So they shed virus in at least viral RNA in saliva and urine. We've detected it in cerebrospinal fluid. And as we mentioned, when we do these necropsies, then we find it in many tissues.
You find it in semen as well?
We have not looked in
testes. Have you looked?
Do we look in testes? It would have to have been in a fetus, so no.
Okay. Yeah, I think this is an important question. I mean, I don't doubt that the CDC should be careful in telling people there may be sexual transmission, but I don't think you can conclude from the data we have so far. There's so few cases, and it could be non-sexual contact, right? So those are important experiments to do.
We are looking at some of the reproductive tract organs right now, and one of our scientists is processing those tissues right now, Dawn Dudley, so she's not able to be here.
So as you know, in mice, you get very good replication in testes, and the CDC paper that just came out this week, they have very nice transmission. An infected male transmit to females very effectively infection. So it is a mouse, I understand, but at least it shows that route can occur. So it would be interesting to get closer to humans.
I think that if anything, we may be underestimating the contribution of sexual transmission, because I think the epidemiologic evidence would suggest that it's intimate partners who have had cases that are clearly non-travel related, and that we haven't seen that amongst others who kiss or have other saliva contact. So I would say the epi evidence does strongly support sexual transmission. I think it's still an open question whether that's a major route or a minor route. I think there's a very open question whether sexual transmission during pregnancy poses a greater or lesser risk than vector transmission during pregnancy. But I think it definitely occurs. And I should say that this is one of the things that we try very hard to do is coordinate with our colleagues at other institutions, experiments, so that we don't are unnecessarily redundant. So our colleague Chris Miller, who we've known for years in SIV, who's an expert in sexual transmission of SIV, is doing intravaginal inoculations with Zika now.
Do you think in the wild monkeys could transmit sexually Zika virus?
If humans can, I suppose monkeys could.
Assuming humans do. I'm not convinced, despite your what you said. I'm not convinced. I think there are not enough cases and there are other issues. But if it's true, why not monkeys? And that could get around. If the mosquito season is low, then it could still be transmitted. That's why serological surveys in Africa would be really interesting on non-human primates. I hope that they can be done, and not just on 30 frozen samples, but an extensive. I know it's not easy to do that, but you have to. Here in the U.S., when the virus comes close, then money gets put up for it. So that's what you have to do. You have to have some threat, because right now we're in a quiet phase, right? There seems to be a diminished global interest. WHO took away the emergency number, the name, and you don't hear as much in the press anymore. Why is that? Do you have any thoughts about that?
Have there been any other big news stories since the emergence of Zika?
It's run its news cycle?
No, I think the new administration and all the concerns about it have sucked the oxygen out of other issues. And it's going to be a big issue, especially with Zika. If the primary effects are overseas, I think those of us who work on Zika, those of us who work on HIV and other global viruses, are really concerned about whether an America First foreign policy is going to extend into research on viruses and long-standing programs like PEPFAR and programs by the CDC to potentially prevent Zika. That's just where we are. But I think that in the case of Zika, my personal belief is that we still have a large Zika-naive population in southern Florida, on the Gulf Coast, and Zika is going to continue to be a story because even a small number of infections in the U.S. in the summer, when the mosquito populations increase, is going to potentially be a major economic liability for those areas. If it gets to Orlando, for example, how many women with families or who are considering families are going to think about changing their travel plans to Disney World? And so I think that even if the number of cases of Zika gets contextualized as, well, this is just another virus that can cause congenital infections like CMV and like the other torch pathogens, Zika is going to punch above its weight economically as long as people are using it to drive their decision making when they're pregnant or thinking of becoming pregnant.
So it's the mosquito that does it, really, because the others are human-human transmitted, and there's more of them than Zika at the moment, but Zika's mosquito, and when you want to go to a holiday, that impacts your ability to do that. What was the question? It slipped out of my mind. What was it? You're just talking about...
I was railing against the administration and global health and that stuff.
That far?
It's gone. All right. So what's next? Can you talk about what's the next series of things you'd like to learn about infection in non-human primates?
Well, I think there's a series of things that we'd like to know. So we've alluded to this idea that there's an extended duration of viremia in pregnancy, and it's going to be next to impossible to really determine what is the mechanism or what are the mechanisms of that in humans alone. And so one thing that we're interested in doing is using the non-human primate model to try to assess what are the contributions of these things that we've been discussing. So is it that that prolonged viremia represents in some way the crossing of the virus into the fetus and then shedding back into the mom? If that's true, can we use that as a kind of clinical assessment tool and say that, as long as a woman who tests positive is within the sort of standard deviation of acute infection, there's an actionable time when we might have therapeutic treatments that could attenuate or prevent transmission to the fetus. And we really need an animal model to assess whether that's the case. So that's one interest. Another interest is how does immunity to related flaviviruses interact with Zika virus pathogenesis? So dengue is very closely related to Zika, as you know, and it's highly prevalent in areas where Zika is emerging. And so there are a lot of people who have been infected with dengue and are now getting exposed to Zika. So we know that antibody-dependent enhancement is a thing in the setting of different Zika serotypes, in different dengue serotypes. And so could pre-existing immunity to dengue affect the trajectory of Zika replication and pathogenesis if you become infected? So we're starting a series of experiments to assess that question here, and I should note that other colleagues are doing that in other institutions.
So that's why you've recently acquired dengue, as you told me earlier, and so you're going to be doing dengue, Zika.
Right. So I personally have hopefully not acquired dengue, but it's in the lab.
Yeah, that's what I meant. So can you, another question has left my mind. What is it? So I suppose you could use, here it is. Can you deplete antibodies and specific immune cells from macaques to see which is important for protection? I don't know if you can do it obviously in mice. Can you do it in yours?
You can do it, and so in fact we and colleagues here at UW have done such experiments in the setting of SIV infection to understand what's the contribution of CD8 positive cells to immunity, say. And so there are treatments that you can do, and I think that's something that has been discussed, but is not immediately on the table. But it's definitely very interesting to understand what are the correlates of protection. What would we be asking a vaccine to do?
We actually have a granted study section today hopefully it's doing okay, but doing the reciprocal experiments where we would take monoclonal antibodies out and passively infuse them back in. So passive adaptive transfer experiments to try to restore protective immunity to an otherwise naive animal would also be possible, either prophylactically or therapeutically.
Right. So I also wanted to ask you about using your model to test vaccines and antivirals, but as you know, apparently now the FDA is not going to require any efficacy in animals before a drug is released into humans. Well, assuming that we're sticking by the old way, could you do that as well?
So absolutely, and I mean, as I was saying, I think that we may be able to define kind of endpoints that you would want to have for a therapeutic to be effective. We certainly kind of understand what endpoints we might want for a vaccine to be effective. We don't want virus replication after challenge. But, there have been discussions, within Team ZEST. If there were therapeutics available today, like what would you do with them? Because if it's just an acute infection that is cleared, then the window for effective treatment might be very small. But if this extended viremia is important for infection of the fetus, then maybe that prolongs the window of time in which you might be able to act to prevent some of the worst impacts of congenital infection.
So if we had a vaccine tomorrow, who would get it?
That's a great question.
Everybody in Brazil?
Pregnant women and women who are planning on becoming pregnant, isn't that the target product profile that the WHO has established? I mean, that's pretty reasonable to me.
But not in the U.S. unless you're traveling to an area that is endemic, right?
Oh, in the U.S., yeah. Well, in the U.S.
It would be a travel vaccine.
A travel vaccine.
But in Brazil and other endemic countries, it would be pregnant women or someone who wants to get pregnant.
Reproductive age women.
Reproductive age. And what about an antiviral? What we have to do to have an antiviral that could be used in a pregnant woman? We have to test it in that population, right?
It would have to be tested in that population. And it's tricky because, in other models where we look at, using an antiviral once a woman has an infection to try to prevent, congenital defects in the fetus, we don't really have a really great model. Looking at CMV, we've used different antivirals to try to mitigate those risks, but it hasn't been as favorable as, we haven't seen as much effect as we'd like. And we're generally not supposed to use them outside of, a clinical trial using different modes of delivery and that sort of thing, but we don't see a lot of effect.
Are there any antivirals licensed for use in pregnant women?
Oh, yeah. We use antivirals like Tamiflu for influenza. That's mostly to try to prevent respiratory morbidity for the mother. But
so
and there are other antivirals we can use, like for herpes.
Right.
We use that to try to decrease the number of lesions so that a woman can have a vaginal delivery because we would try to decrease congenital herpes. But, and there are other viruses that we've used antivirals for, but not for the specific effect of decreasing congenital anomaly risks.
Are there any antivirals that you would use if a pregnant woman was found to be CMV positive?
For the infection itself, we can use antivirals. And there are studies where we try to use antivirals to decrease the congenital infection risk, but that's basically limited to trials.
Okay. So is there anything that we missed that we should talk about?
Well, I wanted to say one thing. So Tom was talking about where we're going next. And one other direction that we're taking our pregnancy model is to actually look at something that is tangible and has an effect, which is fetal outcomes. So not just where the virus goes in the fetus, but are these primate fetuses affected like human fetuses? So when we do get an antibody or a drug that we want to try, does this actually affect the fetal outcome, not just where the virus is? So we're going to be able to look at, do ocular exams, hopefully do hearing evaluations, MRIs.
So far you don't have microcephaly in a primate child whose mother was infected, right?
Right.
But maybe you'll see that if you allow birth and you can see the baby, right?
Or we could see ocular lesions, so retinal lesions.
Sure.
So that's the other marker.
That brings up a good point. The most visible effect in humans are microcephaly, lissencephaly. But do you think there are longer-term cognitive problems that are going to start cropping up years after this initial?
Absolutely.
Right? Yeah.
I mean, there are other congenital infections where you don't detect hearing loss or eye lesions until much later in development, even developmental delays years later.
So you're going to do experiments where you infect and then either C-section or deliver a live fetus and allow them to live for a period and see if you can observe things in them. Can you do behavioral studies in these primates?
You could do this. We don't have those exact plans yet.
So Mary Schneider, who's a researcher here, has spent decades studying fetal alcohol syndrome and sort of cognitive and behavioral assessments that you would do to model fetal alcohol syndrome in macaques would be very similar to those that you would use in Zika. So that was a perfect example of us cold calling someone and saying, hey, you have this related expertise. Would you come and talk to us about what we would do if we ever wanted to do this for Zika? So she came out and gave a talk in front of our whole group and shared that.
Also, the two of you, the two docs, what do you think if a baby is infected at birth or within the first year or so, the brain is still developing? Is there potential for infection and neurological problems there, you think?
I guess there's theoretical.
We haven't seen it, I know.
Right,
but
we haven't seen it. And babies in the area live with dengue and West Nile infection, and we haven't seen that with related viruses. I realize we haven't seen microcephaly with dengue or West Nile either. But we haven't seen that yet. I know there are lots of trials down there looking at that. So we'll have to, I think, wait and see.
And there was some evidence, I guess, from the Yap Islands that when babies were vertically infected right at the time of birth, they weren't really seeing, a lot of adverse outcomes. But also that epidemic was only a few years ago, and so a lot remains to be seen, as she said.
Have you had travel cases of Zika in Wisconsin? Yes.
Yes.
How many?
When I last looked in November, I believe, I don't remember if the number was like 48 or something like that. And have
babies with birth defects been born to some of those mothers, do you know?
There have only been a few women who were pregnant in the state of Wisconsin who've had Zika. And to the best of my knowledge, most of those have ongoing pregnancies. And I haven't seen anything published on the outcomes within this state.
Do you have Aedes aegypti here in Wisconsin? No, probably not. Probably we don't have it in New Jersey or New York either. So that's good. So did I forget anything? Is there something tough I can ask them that you can feed me? No?
Can I say, we could have had many guests on the show. We only had space for 4, so I want to say thank you to all the Team ZEST members, so Don Dudley, Matt Eliotto, Christina Newman, everybody that could have been on in our place here.
Yeah, she wanted to have 50. I said, no, I don't have enough microphones for that many people. But some Team ZEST here. You're all Team ZEST? Not all, right?
Not all.
Some,
no,
right?
Some.
All right.
Some.
ZEST. That was a soap, isn't it, ZEST?
Yes.
Is it?
Yes.
Maybe that'll be the show image. No, just kidding. All right. That's TWiV 429. All these episodes you can find at microbe.tv/TWiV. You can find them on iTunes, and if you listen on your phone or tablet, there are apps that you can use to subscribe, download for free, and so forth. And if you have any questions or comments about this episode or any of the others, if you send us your question, we'll pass them on to our guest, TWiV at microbe.tv. And consider supporting our work. You can go to microbe.tv/contribute, and you can help us so that we can travel, do more shows, and so forth, pay for our costs. We have a Patreon account. We have an Amazon affiliates account. Cash, diamonds, gold, platinum, whatever you want to donate can help us out. Microbe.tv/contribute. I want to thank all of my guests today. David O'Connor, thank you again for returning. That's your second visit. Think there'll be more?
I would hope so. Always happy to talk.
Thank you very much. Emma Moore here at the University of Wisconsin's Pediatric Infectious Diseases. Thank you, Emma.
Thank you.
Did you enjoy TWiV?
I loved it.
And thank you for inviting me and hosting and all of that. And look forward to the rest of my minute. Katie Anthony and OBGYN here. Thank you so much.
Thank you.
It's your first podcast.
It is.
Isn't it cool?
It's fun.
So our listeners are all sorts of people interested in science. And it's really important to get scientists and physicians on the show like this. That's why I like doing this.
I love it.
And I will do this into the sunset. And finally, the man who didn't leave, Tom Friedrich.
Didn't leave.
Thank you very much.
Thank you.
And you see,
Thank you for having me back despite my abrupt
yes, but you made a wonderful contribution by staying. It's really good. You have a lot to say, and I'm sad you left last time, but thank you so much. Now we made it up, and it's all under the bridge. So thanks, everyone. Thanks for the audience for being here as well. We have a nice little interested audience who I assume are fans. I also want to thank the sponsor of this episode, Blue Apron. I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.